So this is the procedure by which we produce monoclonal antibodies. First, a mouse is exposed to the non-self antigen. The B cells in the mouse will produce a mixture of antibodies. We extract this from the spleen of the mouse. We mix these B cells that are activated by the antigen that we extracted with cancer cells. This allows them to divide rapidly because cancer cells divide rapidly and uncontrollably. And this happens by the addition of a detergent that breaks down the membranes of the B cells as well as the cancer cells. This allows them to fuse together, and the fusion of these two is called a hybridoma cell. Hybridoma cells are basically hybrids of cancer cells and B cells. They are cells that have uncontrolled and rapid division and that also produce antibodies. Then we separate these hybridoma cells under a microscope. We choose the ones that are the best and we culture them. Each one of them is allowed to form a clone, and then each clone is tested to see if it's producing the required antibody. If it does produce the required antibody, then this clone is a successful clone. We grow it on a large scale, and then we extract antibodies from it, and there we go. We have monoclonal antibodies. Before we use them on humans, we have to put them through a process called humanization, which means we modify them to make them more like human cells. Because remember, these are mouse cells, which means they are going to be recognized as non-self. And we don't want our body cells recognizing them as non-self and attacking them. We need them in order to help us overcome something. We don't want more problems by having our own immune cells attacking these monoclonal antibodies. So this is how we make monoclonal antibodies in the lab.